Thanks, Zaza, and thank you, Bruce, very much for the opportunity to present and to be here today. I am one of those chemists and physicists who was recruited through the mechanisms that Bruce talked about. And my lab, which is split between MIT and the Ragon, focuses on developing technologies to understand how immune responses are structured. So as just came up in Zaza's talk, and as I'm sure you're well aware, we are complex multicellular organisms. Within our bodies, there are systems that are in turn comprised of organs, which within them have tissues, which are made up of sort of the atomic units of our bodies, which are cells. In the past, what has traditionally been done is we take our favorite cell, like the CD8 T cell, we take a population of it and we characterize it deeply. What this does, though, is it doesn't give us a sense of what each individual cell might be doing, and it doesn't give us a sense of how that cell might be working with all of its compatriots to drive a response as a whole. What my lab has been working on over the past several years is developing technologies that will let us take very complicated cellular mixtures, profile them deeply, and using the power of genomics and computational approaches, identify groups within these cells, so based upon the things that they're doing, and then create a parts list and also an understanding of what are the characteristics of these various elements. And so what attracted me to the Ragon originally was this idea that I could take these technologies that we were building as engineers and apply them to real-world problems and potentially have impact. And so I got excited when I started talking to people like Doug Kwan, who's our MC for today, about using these to figure out what the acute targets of HIV infection were, or to work with Sarah Fortune to look at granulomas to try and figure out what made a granuloma restrict bacteria, or what made it permissive so that we had outgrowth. But what I realized when I started talking with them was that the technologies that we had would not actually solve the problems that they needed. We didn't have things that were scalable, so we could never work on these complex systems given heterogeneity among individuals. We had to run them in our lab, so we weren't going to bring them to the hospital suite or to the BSL-3. We needed to be more efficient so we could be better at using the samples that we got, and we needed to drive down the cost so that we could really scale up and have impact. And what this led us to do is to go put back on our engineering hat with our collaborator Chris Love over at MIT, who's also an associate member here at the Ragon, and to really engineer all of these complicated systems into this very simple microwell chip that works based upon gravity. And so to give you a sense for how this works really quickly, here's a little simple diagram, or actually I took it out. I'll just tell you that the way in which it works is that it takes these uniquely barcoded beads, couples them together with cells in these microwells, and then we can basically capture all the information from the cell right here, and then figure out what belongs to whom based upon these sequences. So we spent a lot of time testing in our labs to prove that we could get good capture in terms of numbers of genes and information per cell, and do as well, if not better, than the commercially available variants. We worked with partners all around Boston to make sure that we could work with every clinical specimen that we might need to be able to work with, from blood to pinch biopsies from the gut to even possicellular fluids like cerebrospinal fluid that you'd never think about working with. And we pretty much figured out that we could work with just about anything. But then we took the real test and partnered with some of our collaborators at KRIF, now ARI, to actually go and try and test these on some of the field samples that were most important, looking at HIV-positive lymph nodes versus ones from healthy donors, and beginning to see the shifts that occur as a result of infection, as well as which cells are actually actively harboring virus. And in doing all of this, we built incredible partnerships with people on the ground in South Africa. And one of the things that's the most rewarding to my entire lab throughout this has been the ability to connect and to get to know not only the nature of the problem so we can engineer solutions to it, but also to really understand the faces behind the numbers that have been quoted today, because it really changes the motivation when you know who you're doing this work for. And it's actually done that so much so that my entire lab has transitioned from a lab that worked primarily in isolation when I was a graduate student to one where we now work around the globe with hundreds of partners on all sorts of infectious diseases. And what we really have now are these beautiful molecular and cellular maps of what's happening in different disease settings that we're trying to use to identify common unique themes. Really the goal of all of this is to go identify therapeutic and prophylactic targets that we can start to work on with partners like Dan Baruch, who's going to come up next and tell us about some of the initial vaccine studies.